Chapter 5, Topic 5.3, Viral Replication. We will be covering the multiplication cycles in animal viruses, damage to the host cell and persistent infections, viruses and cancer, viruses that infect bacteria, and lysogeny. At the end of this recorded lecture, you should be able to diagram the 5-step life cycle of animal viruses, explain what cytopathic effects are, discuss both persistent and transforming infections, provide a thorough description of lysogenic and lytic bacteriophage infections. So let's talk about how viruses make more of themselves. The first step in viral replication is to grab a hold of a suitable host cell. Viruses don't have hands. Instead, they have spikes, which interact with a molecule on the cell, the cell receptor. Now, the cell receptor is not there to let viruses in. Cells don't want viruses to come in and take them over. The receptor is doing another job for the cell, and the virus uses it for its own nefarious plan. The spike and the receptor fit together like a lock and a key. Actually, it's more like the virus picking the lock on the city gate. Now that the virus has absorbed to the cell, it now needs to get its genome into the cell. Different viruses do this in different ways. If the virus has an envelope, like in this diagram, it can fuse the envelope with the cell membrane, spitting the capsid into the cell. Or, if it doesn't have an envelope, it may inject its genome into the cell, or it may trick the cell into bringing it in, like this enveloped virus did. Any way that the virus can enter the cell is called penetration. Once inside the cell, if it hasn't already done so during penetration, the virus needs to get rid of the capsid. It's done its job of protecting the virus while it was out in the cold, hostile world outside the host cell, but it is unneeded now. Getting rid of the capsid is called encoding. Once the genome is free within the cell, it goes about making viral proteins and copies of its genome. So here, in this example, we have a positive sense RNA genome, so we can immediately start making new viral parts. It makes a negative copy so that we can make more positive copies of the genome. This is called synthesis. Now, if the virus has a DNA genome, the genome goes into the nucleus of the animal cell, because that's where all the enzymes for working with DNA are in the cell. Those enzymes make more DNA copies of the genome, and they make RNA copies, mRNAs, that go out into the cytoplasm where they code for viral capsid proteins, viral enzymes, and so on. If the virus has an RNA genome, it usually stays in the cytoplasm like in this diagram. So to recap synthesis, whether it's an RNA virus like in this example, or a DNA virus, synthesis involves making viral proteins and new genomes, because no matter what the genetic configuration of the virus is, each new virus will need a copy of the genome. Once the host cell has made all of the necessary parts, new viruses begin to assemble. Capsid proteins form new little capsids, genomes are stuffed into the capsids. For enveloped viruses, spike proteins are transported to the cell's cytoplasmic membrane. Now we have a new bunch of little viruses. It is time to send them out into the world to infect new host cells. The virally infected cell may pop or lyse, releasing the new viruses. Some enveloped viruses, like in this diagram, bud out of the cell, picking up their spikes and their envelopes on their way out. So the 5 steps in viral replication are adsorption, penetration, encoding, synthesis, assembly, and release. Understandably, this process damages the host cell. While it is making proteins and nucleic acids for the virus, the cell falls behind on making its own proteins, nucleic acids, carbohydrates, and lipids. Physical changes in the appearance of the host cell that can sometimes be seen using a microscope are called cytopathic effect, or CPE. CPE varies from virus to virus so that we can identify the virus based on the CPE it causes. Here's an example of normal cells in cell culture, and here you can see they're kind of rounding up and they're clumping together. That would be considered CPE. Here we've got an example of where you can see with your eyes areas where the cells have been killed. We call these plaques. Some viruses are slower growing and establish a persistent infection of the cell, or a carrier state. Depending upon the virus, the cell can produce viruses for years before the cell dies. HIV is an example of this. HIV is a retrovirus, which means that it starts out with an RNA genome, but its enzyme, reverse transcriptase, makes a DNA copy of the genome, which is then inserted into the host cell genome. This is called a provirus. Once there, the provirus codes for more viruses, which leave the cell without killing it. This is one of the reasons that HIV has proved so difficult to cure or even treat. Other viruses, such as herpes zoster virus, the cause of chickenpox and shingles, go into a chronic latent state. Between bursts of virus synthesis, assembly, and release, the genome hangs out in a latent or inactive state, hiding within the cell. Then it bursts into activity when the immune system isn't looking, and you get shingles. Some animal viruses permanently alter the cell's genome, causing the cell to become cancerous. These viruses are oncogenic, and their effect on the cells is termed transformation. It is estimated that 20% of all human cancers are caused by viruses. Just think of human papillomavirus and cervical cancer, or hepatitis B or C and liver cancer. Viruses most often cause cancer in 3 ways. I'll leave you to read about these 3 ways in the textbook, but it all boils down to this. Viruses provoke uncontrolled cell growth. Uncontrolled cell growth in a multicellular organism is cancer. Bacteria are also plagued with viruses. We call these viruses bacteriophages, or phages for short. Phages have the same step in their replication cycle that animal viruses do. Let's go over this figure to review those steps. Absorption, penetration, duplication or synthesis and assembly, maturation, which is also assembly, and then release of the cell. The main difference between animal viruses and phages, besides their target host cells, are that phages are limited in how they get in and out of bacterial cells. Because of the bacterial cell wall, they can't bring in the phage whole, nor can the phage butt out of the cell. So phages must penetrate by injecting their DNA into the bacterial cell, and to be released, they must lyse or pop the host cell. Some phages set up a type of latent infection. Rather than taking over cellular processes right away, the phage inserts its DNA into the bacterium's DNA. There, it hangs out as a prophage. It replicates every time the bacterium divides, so it spreads within the bacterial population. Some lysogenic phages bring gifts of antibiotic resistance or toxins to its host cell, making the bacterium where it is a prophage better than it was before, stronger, faster, etc. That is good for the bacterium, but it's usually bad for us humans. One example of a bacterium that is made worse for humans by lysogeny is the bacterium that causes diphtheria. Without the prophage, Corynebacterium diphtheriae is non-pathogenic. When it has a prophage that carries a toxin gene, then C. diphtheriae causes, well, diphtheria. Suppose the bacterium with the prophage is damaged. Suppose I applied a disinfectant and popped holes all over the cell, in its cell wall and in the cell membrane. The prophage says, "Whoa, buddy, this boat is leaking. It's time to leave this ship." And it moves into lytic phase. It kicks into synthesis and assembly phase before the bacterium dies. Then it pops the bacterium so the new phages can find a new, non-leaking, non-dying home. And so the cycle continues. That's it for this topic. You should now be able to diagram the 5-step life cycle of an animal virus, explain what cytopathic effects are, discuss both persistent and transforming infections, provide a thorough description of lysogenic and lytic bacterial phage infections.